All right, guys. Quarantine video number 3. The last video of this unit, and it will be on the brain. So pulling it up now. We'll get going. All right. So this is kind of the culmination of what we've been talking about here for the last couple lectures, bringing the neurons of the body, not all the neurons, but the majority of our neurons, 85 billion of them to make up our fairly small brain, only about 3 pounds, but it's super important. Everybody's aware even before taking anatomy, physiology style classes. Allows us to do things like solve equations, feel hungry, laugh. Those are all pretty straightforward, simple things that we experience on a daily basis, and they all are things that occur in different regions of our brain. Aside from the neurons that I mentioned that make up our brain, there's upwards of 50 trillion neuroglia, those supporting cells, so like the astrocytes and the microglia, ependymal cells, so on and so forth. And we can describe it as the control center for registering sensations, correlating them with one another, and with stored information, as well as making decisions and then taking actions. It's responsible for our intellect, our emotions, our behavior, and our memory. And there are kind of 4 major parts to the brain. The inferiormost portion of your brain is what we call the brain stem, and that includes 3 major structures: your medulla oblongata, the pons, and then the structure that's kind of split in half here, what we call the midbrain. Okay. Right behind that, so posterior to your brain stem, is the major structure known as your cerebellum. Cerebellum is a structure that's the second largest part of the brain, largest being the cerebrum, which is by far and away the largest, is all of this up on top. And in the middle between your brain stem and your cerebrum is what we call the diencephalon. The diencephalon includes the thalamus, this kind of egg-shaped, oval-shaped structure, the hypothalamus, and then the structures that make up the epithalamus, which includes what we call the pineal gland. Aside from that, there are many other structures that are associated with the brain. Pituitary gland is something that releases hormones. It is acted upon by the hypothalamus. We'll talk about the pituitary gland in the next unit. We're going to talk about these little cavities in this set of canals that allow for the flow of cerebrospinal fluid to occur throughout the brain and down into our spinal cord. We'll talk about the kind of structural parts to the cerebrum and things like this, what we call the cingulate gyrus, and this, which is called the corpus callosum, this, which is the septum pellucidum. Talk about the gray matter and white matter in the brain and spinal cord. Or we have talked about those, but we'll talk about them kind of in greater detail with the cerebrum and especially the cerebellum. This white thing here is actually known as the arbor vitae, because if you turn the page sideways, you can kind of see a tree-like structure, and that's what arbor vitae actually translates to mean, tree of life. So those are kind of some most important parts of our brain. We're going to talk about them kind of individually and learn what they do and kind of a little bit more about their anatomy here in the next several slides. Before we do that, I want to talk about the meninges that we talked about in our lecture on the spinal cord. The same 3 meninges as we talked about protect our brain as they did around our spinal cord, and it's dura, arachnoid, and pia mater. Same situation, superficial to deep. The dura mater is the tough mother, as we learned, made up of 2 layers, an outer and an inner layer. If I zoom in down here at the bottom, looking at kind of just a small segment of the brain, we can actually see the scalp or skin. Then you have the bone, in this case, 2 different parietal bones, because we are split right down the middle. And then you can see in green here is our 2 layers of the dura mater. What we call periosteal layer is the superficial layer. Makes sense that the periosteal layer is superficial because if you remember, the wrapping around our bones is known as periosteum, and that's right where the periosteal layer is connected to. The deeper layer is known as the meningeal layer, okay, and those are the 2 layers of the dura mater. What we do not see that we talked about last time is a space between the cranial bones in the dura mater. We saw that with the spinal column and the spinal cord, and that was called the epidural space. There is no epidural space surrounding the brain. And there is a couple spaces, though, in various places associated with the dura mater, and these are called dural sinuses, which are channels within the dura mater that contain venous blood that returns from our brain to what we call the internal jugular veins. So after blood has flowed into the brain and fed the brain, it then needs to return back to the heart, just like blood anywhere else in the body. And so these sinuses allow for the blood to kind of collect and then eventually drain into what's called your internal jugular vein, which is the main blood vessel on each side of our brain that drains our brain. Okay, and we can see here is an example of one in this kind of triangular shaped area is the superior sagittal sinus. Over here, this is actually from a cadaver. We can see the same thing. It's not the perfect triangle that we saw over here, but we can definitely see there's a space in there. There's also associated with the dura mater 3 major extensions that help separate the brain. There's what we call the falx cerebri, which we can see here and over here. The falx cerebri, also shown down here, is what separates the cerebrum into its 2 halves. The cerebrum, once again, is that big, large portion of our brain, and it's actually made up of 2 what we call hemispheres. And penetrating down to help separate them from one another is this falx cerebri. Now, similar to that, and spelled very similar but very different, is the falx cerebelli, which separates the 2 halves to your cerebellum. So cerebri for cerebrum, cerebelli for cerebellum. And then the final structure is this one here, the tentorium cerebelli, which is what separates the cerebrum from the cerebellum. Okay. All right, moving on to the arachnoid mater. Arachnoid mater, remember, this is the spider mother, is very thin in our picture here. This is the kind of pink in our 2 halves of this picture. The arachnoid mater, you can see over here in the subarachnoid space. I'll talk a little bit more about that stuff. Kind of looks like a spider web, and that's what we were talking about the other day. Associated with the arachnoid mater are these things that kind of look like little trees, these little pink extensions from the arachnoid mater, penetrate out into the dura mater, especially into those dural sinuses. And what they do is transport cerebrospinal fluid from that subarachnoid space to the dural sinuses and help return that cerebrospinal the fluid in our cerebrospinal fluid back to our blood, because we're always producing more, and so we're losing some, but we're gaining some all the time. And speaking of the subarachnoid space, this contains blood vessels and helps circulate the cerebrospinal fluid, and you can see very pronounced space over here in our real deal picture. And then finally, the pia mater is the delicate mother, and that would be this kind of red right there covering the surface of the brain, and it functions to carry blood vessels and provide nourishment to the nervous tissue. So those are the protective structures. Okay. Also, something that does help protect our brain is cerebrospinal fluid, which I was already mentioning. CSF, as it's abbreviated, located in the subarachnoid space, but also within cavities in the canals of the brain and spinal cord. And in our brain, we are going to focus on these 4 large, what we call ventricles. Okay. Before we do that, I just want to talk a little bit about cerebrospinal fluid. It's a clear, colorless liquid, contains glucose, proteins, lactic acid, urea, cations, anions, and some white blood cells. So there's a lot of nutrients. There's some waste. There's cells. So it's kind of a hodgepodge. Okay. But each of those is going to have a specific reason for being in there. Function of the cerebrospinal fluid is, in our ventricles, it functions to absorb physical shocks to the brain. It kind of actually allows our brain to kind of float, if you kind of think very not so literally, but that just helps prevent things like concussions. More so, it helps distribute nutritive materials to and remove waste from our nervous tissue, and provides a chemically stable environment for things to occur naturally and normally in our brain. We learned about the blood-brain barrier back in our first lecture of the unit. Formed by the astrocytes, is the separation of our blood plasma from capillaries and the cerebrospinal fluid in the ventricles. Okay, so let's get into the 4 ventricles. Okay. Here we have an illustration to kind of show them. It's a lateral view. You would not be able to see these in a natural lateral view, but you can because they have just kind of projected them to the surface. So that's why they kind of look a little bit translucent a little bit, just kind of showing you where they would be at in the entirety of the brain. The two largest by far and away are these kind of C-shaped lateral ventricles. There's one on each side, and I really like these pictures at the bottom to show from above. This is a cadaver that's had the top cut off, so transverse section, and then this is a frontal section where the front of the brain's been cut off, and you can see the two big chambers known as the lateral ventricles. There's one associated with each of our hemispheres of the cerebrum. Okay, now separating them, and you can kind of see it right here. You can definitely see it right here, is a membrane that I pointed out on that first page called the septum pellucidum. If I go back to that first page, that would be this little sheet right there. Okay. And so that helps to kind of connect them, but also keep the fluid where they're supposed to be. The next structure, the next ventricle, is the third ventricle. It's immediately inferior to kind of the upper part of the C, if you think of it, like a C for the two lateral ventricles. And we can't see them here, but we can see the lateral ventricle, or excuse me, the third ventricle right here. What we can't see in this illustration, or this picture, I should say, it's not an illustration, would be the presence of two little tiny channels that allow the cerebrospinal fluid from the lateral ventricles to flow down into the third ventricle. And you can see just right, barely anything whatsoever, just a little channel right there. It's called an interventricular foramen. And now up here in my text, I have them listed as interventricular foramina. Once again, foramina is plural, whereas foramen is singular, and that was only showing you one, but there are two because there's one from each of the two lateral ventricles. Okay. From there, cerebrospinal fluid will flow down through this chamber, or into this chamber, I should say, the chamber being the fourth ventricle, but it does so by a tube known as the aqueduct of the midbrain, or what we call the cerebral aqueduct. And if I go back to this first page, okay, this channel here is that cerebral aqueduct, or aqueduct of the midbrain, and you can probably figure out why we call it that, since what splits the midbrain in half. And then here, right between the cerebellum and the pons, is what we call the fourth ventricle. It's a diamond-shaped space, narrows caudally to become the central canal, which is the chamber that's found inside of the middle of our H that we learned about in our last lecture. And the important thing to understand is that the flow of this fluid is not going to be like what we see with like a pond, where it's just stagnant, staying in the chambers. Instead, the fluid that's in the lateral ventricles then flows down into the third, and then the fourth, and then possibly down into the central canal, or it could go to the subarachnoid space and then be returned to the blood. And that's because we're always producing more. And by producing it, I mean it's done by way of blood capillaries called choroid plexuses. And these are networks of blood capillaries that pull the water and the nutrients and stuff from the blood and push it out into those ventricles, forming cerebrospinal fluid. And it allows those ependymal cells that we talked about to secrete the cerebrospinal fluid. And we can see that here in this illustration. And an example of a choroid plexus, here's the blood vessel, water and some stuff is removed, goes through the cells here called the ependymal cells, and cerebrospinal fluid is created. Okay, so it's a nice flow of fluid, which is very, very cool. Okay, now that we've kind of got some of the introductory stuff out of the way, and now we can actually get into talking about the different parts of the brain. So what we're going to do is start at the bottom, just work our way up to the top, beginning with the most inferior portion of our brain, the medulla oblongata. You'll probably just hear me call it medulla most of the time. It's about 3 centimeters in length, so it's about an inch, a little over an inch. Superficially, it just looks like an extension of the spinal cord, but it's just a little bit slightly wider, excuse me. Didn't include a picture or that same picture that was on that first page, but I did include a couple pictures to show off some different things. This is an inferior view of the brain. So what we've done, pop the brain out, flip it upside down. And we can see down here at the bottom, this would be the spinal cord. And just above it, which is kind of hard to imagine that this is above it, but remember you're thinking this is upside down, are the parts of our medulla oblongata. And this is a transverse section, and I'm going to talk about these things called pyramids here in just a little bit. Yeah, but first, let's just talk about what the medulla does. Okay, within the medulla are nuclei. If you think back to that first lecture, we learned that nuclei are collections of neuronal cell bodies in the central nervous system. So these nuclei are several neurons in their cell bodies that are components of sensory pathways for gestation, which is a fancy word for taste, audition, which is a fancy word for hearing, and equilibrium, a fancy word for balance. So 3 out of our 5 special senses are going to involve movement through the medulla. There's also nuclei associated with other sensations, touch, pressure, vibration, conscious proprioception, which is kind of a fancy term to describe your sense of balance, position of joints, stretch of muscles, tension in tendons. It's a fairly complex thing, but it's a very important thing at the same time. Probably the meat and potatoes, I would say, of the medulla, what it's most important for are these 3 bullets here, because these are life and death type things. We can classify our medulla as a major regulator of our entire cardiovascular system. And there are 3 components to our cardiovascular system. You got the blood, which flows through our blood vessels, and then the heart, which pumps that blood out into our blood vessels. Well, our medulla controls how fast and how hard our heart beats. It has what we call the cardiac center that does that. And then the vasomotor center is responsible for routing blood and controlling blood pressure by way of what we call vasoconstriction and vasodilation, which is where we can allow blood to travel more or less through certain blood vessels, which is a very important thing. Okay. And then the third kind of most important thing is its role in breathing. Within the medulla is what we call the medullary respiratory rhythmicity area. So you literally just take off the RY here in the first word, it tells you who's involved. It's gonna be the part of our brain that helps adjust our rhythm of breathing. So how often we breathe. If we breathe more rapidly or more slowly, your medulla is involved. Other things that it does involve some of the more important, but not nearly as kind of life and death things: vomiting, swallowing, sneezing, coughing, and hiccuping. And the reason I included this picture, and you can actually see them here, these 2 bulges, and I pointed them out just a second ago. These structures are called pyramids, okay, which is where a process called decussation takes place. Okay. But these bulges on the anterior aspect of the medulla are formed by corticospinal tracts that pass from the cerebellum down the spinal cord. These corticospinal tracts control voluntary movement of our limbs and our trunk. And, if you look in this illustration, you see where they form kind of an X, and that's why it's labeled there, decussation. When those signals come down from the brain, they crisscross from one side to the other, and it explains why if I wave my right hand, for example, the left side of my brain is actually in charge of doing that, and vice versa. That's for motor signals, but that's also the same for a lot of the sensory signals by way of those ascending tracks. Not every signal, only about 90%, but that's by far and away the majority of them. And that explains why we have that opposite side feature that many of you probably already knew about. Okay, last thing I want to talk about, and this illustration really shows it, are these things that are kind of golden color, which are the just basically the trunks of the 12 pairs of cranial nerves, and which we've mentioned at various points in this unit are nerves that are attached to the base of the brain. You can see a lot of them are attached to our medulla. And in fact, 8, 9, 10, 11, and 12 are the pairs that are associated with our medulla oblongata. We can actually see a couple pairs of them here. Actually, all 5 of them here in our picture on the right, but these pictures are not something I would really focus too much time on. Not something I would really ask you to know. All right, the pons, which is just above the medulla, is kind of known as the bridge, and that's actually what pons translates to mean. And that's because it connects a lot of different parts of the brain with one another. quite a bit fatter, bulges a lot more anteriorly than the medulla. It has white matter that include tracts conducting signals from the cerebrum down to the cerebellum and medulla, tracts that are carrying signals up to the thalamus. So there's nuclei, there's sensory and motor tracts. It's a lot of different things. Okay. Cranial nerves 5, 6, 7, and 8 are involving the pons and are connected to it. Probably the most important thing that I always tell students to remember is this second bullet. What it does within the pons is what we call the pneumotaxic and apneustic areas, which are another set of parts of the brain that help control our breathing. I won't talk about what specifics they do, but they help with the medulla in regulating and controlling breathing. Other things that controls vestibular nuclei, components of the equilibrium pathway from our inner ear to the brain, so it's involved with balance, and signals for voluntary movements from motor areas of our cerebral cortex are relayed through nuclei in the pons on the way to the cerebellum. Okay. Sitting above the pons is the last segment of the brainstem, what we call the midbrain or mesencephalon. It's a short segment of the brainstem, and it functions to connect the pons to the diencephalon. The aqueduct of the midbrain or cerebral aqueduct we talked about earlier passes through it, and a lot of different things take place as a result of our midbrain. Anterior part contains paired bundles of axons called cerebral peduncles. These are axons of motor tracts conducting nerve impulses from the cerebral cortex to the spinal cord, pons, and medulla. Probably one of the most important things it does is helps control awareness of pain, which is super important, and actually is a really good thing, despite the fact that most people would say pain is bad. Pain is actually really good for us because it tells us when something's wrong. It tells us when damage is occurring to the body. If we didn't have pain, we could be damaging our body and we would just never know it. So midbrain is involved with that. A lot of things with vision, tracking moving objects, blinking, focusing our pupils, focusing, dilating, and constricting our pupils, turning your eyes and head in response to, like, if somebody's in your periphery or something's moving in your periphery. That way, if something's coming after us, we're ready for it. We look that way, so we got a good sight of it, because our clearest vision is always straight ahead. You don't read by putting your notebook off to the side. You put it in front of you. So by looking in that direction, you can see what's going on. It's also involved with auditory things like the startle reflex. Like if you hear a loud noise, you'll kind of crouch down, and you'll kind of turn your head and trunk that way. So that's another protective mechanism. Contains several nuclei, one of which is known as the substantia nigra, which is illustrated here in our picture. In here, kind of more internally, portions of the cerebral peduncles that we can see. This releases dopamine involved in the subconscious control of body movements, and it's the loss of these neurons that's associated with people that have Parkinson's disease. Parkinson's, if you're not familiar with it, is a disorder characterized by people that just have an inability to relax their skeletal muscles, so they're constantly just kind of doing these kind of motions, can't stop from doing that, and it interferes with just like writing your name and getting dressed and just doing any simple task. Typically it affects people in their 60s. It's very debilitating. Not a whole lot can be done for it. Probably the most well-known person with it is Michael J. Fox, which was he was a prominent child actor and adult actor in the 90s, late 80s. You ever seen like the Back to the Future movies? That's what he's in. There's some really other good ones out there, but he was like in his 40s, I think, when he was diagnosed with it. Pretty much kind of shut down his acting career for quite a while, but he's been in some things recently here and there, but just not nearly as prominently as he once was. Also shown in this illustration are red nuclei, which are these things that are nice and red, fancily known, or fancily there in the picture. These are sites of synapses between axons from the cerebellum and the cerebral cortex that help control some voluntary movement of our limbs. And then cranial nerves 3 and 4 are attached to the midbrain. As far as the images goes, this is the same one that was on that previous page, where you can see things like those cerebral peduncles that I pointed out. Not something you would really focus too much on studying. Okay, now if I were to go back to that first page, you would not be able to just kind of locate, and I didn't even point it out or say anything about it. This next set of structures, I'm going to talk about what we call the reticular formation. This is a cluster of gray matter that's kind of interspersed or sprinkled in within white matter of our brainstem, as well as certain regions of our spinal cord, diencephalon, and cerebellum. So it's just kind of this general area where you just got kind of gray matter sprinkled in. Okay. Part of it, what we call the reticular activating system, consists of sensory axons that project out to the cerebral cortex, which is what these arrows are illustrating. Cerebral cortex is the very outer portion of our cerebrum, where the highest level of thinking takes place. But a lot of things involve our reticulating activating system, or RAS. Possibly the most important of which is consciousness, which hopefully you guys are all demonstrating right now, a state of wakefulness in which an individual is fully alert, aware, and oriented. If you inactivate the RAS, it can produce sleep, which is defined as a state of partial consciousness from which an individual can be aroused. Aroused is just a fancy word for waking from sleep, meaning our RAS is involved with that. Okay? Things that can trigger you to wake up, like loud noises, light hitting your eyes. That's why darkness helps us sleep. Things that won't activate the RAS to make us wake up, would like sense of smell. And that's why we have to have smoke detectors. We can't smell smoke, and we can't smell anything. We can hear things. We can feel when people touch us. It's just kind of a unique situation, and that's why we have to have smoke detectors in our house. Pretty much similarly to sleep is coma. Okay, coma. The difference is if you damage the RAS, you can get a coma, and this is where you cannot be aroused. That's the difference between sleep and coma. You can't just wake someone up by throwing water on their face or something like that. Other things that helps maintain attention, like concentrating on a single object or thought, helps with alertness, and prevents sensory overload by filtering out unimportant sensory information. So a lot of times I teach this in a room that can be quite noisy outside with other students and that kind of stuff, and I always try to point out that there's been students talking outside or cars driving by, and I ask students a lot of times if they heard them while they're listening to me. Most of the time they say no, which makes me happy that they are preventing that sensory overload by just keeping that stuff blocked out. And then finally, it helps maintain muscle tone, which is a slight degree of involuntary contraction, even when we feel like our muscles aren't completely at rest. So even if you feel like you're laying down and your body is just completely rested, your muscles are not doing much, not doing anything, they really truly are. And that's because of muscle tone as a result of the RAS, which is cool. All right, moving on from the brainstem, we'll move to the cerebellum, largest part of the hindbrain, second largest part of the brain. I've got several illustrations here to show parts of it, and actually a view from a cadaver in a mid-sagittal cut. So just like what was on that first page, just kind of zooming in at the bottom portion of it. There's a lot of similarities that exist between the cerebrum and the cerebellum, aside from just having similarly sounding names. They're both made up of two hemispheres, a left and a right. We call these cerebellar hemispheres, whereas the cerebrum has what we call cerebral hemispheres. Both of them have kind of outward folds on the outside to increase surface area, which allows for more and more and more neurons. In fact, there's an incredible number of neurons in our cerebellum. Even though our cerebellum only consists of 1/10 of the brain mass, nearly half of the neurons in our brain are found here. Let's see, reading through. Lies posterior to medulla and pons, inferior to the posterior portion of cerebrum, deep groove known as a transverse fissure along with that tentorium cerebelli that we talked about earlier, separates the cerebellum from the cerebrum. Two hemispheres are connected by a bridge called a vermis, which we can see here. This is a view above looking down at the two hemispheres. Here's an image looking up, and we can see the vermis again. Just like the cerebrum, which we'll find out, the cerebellum is then subdivided. Both hemispheres have what we call lobes. Our cerebellum, each hemisphere has 3 lobes, an anterior and a posterior lobe. And then a lobe I could never pronounce, so I just call it the F lobe. Okay, and I can't even try to say it. Okay, but that F lobe contributes to equilibrium and balance, whereas the anterior and posterior lobes govern subconscious aspects of skeletal muscle movements. Those folds that I was talking about earlier, and you can see them here in all of our illustrations, are called folia. Okay, and then the white matter, because the folia is gray matter, is what we call the arbor vitae. I talked about that earlier. Okay, and going along with that tree theme, if you think of trees, what goes on trees are leaves, okay, which, if you've ever heard the term foliage, refers to the leaves of trees. Like, I've, hear people talk about beautiful fall foliage when the trees turn orange in the fall. So folia would be like the leaves that are on the branches of our tree. So the white matter being the arbor vitae, and then the gray matter, those folds are called folia on the outside. And so there's also, and you can't see them, there's kind of 4 little dots of more gray matter, what we call deep cerebellar nuclei, which give rise to axons carrying impulses from our cerebellum to other brain centers. Our cerebellum is connected to the brain stem via 3 pairs of cerebellar peduncles, which are bundles of white matter consisting of axons conducting impulses between the cerebellum and other parts of the brain, helping to coordinate movement, regulate posture, and balance. And you can see those superior, middle, inferior in our various illustrations. And I'm not going to read through because there's a lot of information in here, but they all have their own specifics. And overall, what our cerebellum does is to help evaluate how well movements are initiated by motor areas in the cerebrum and how they're actually being carried out. So what that means is when I wave my hand, for example, that signal's not coming from my cerebellum to do that. The cerebrum is where that signal is coming from. But there's also signals at the same time going to my cerebellum and signals coming back from my arm to the cerebellum. That's basically making sure that that signal was being carried out correctly. So I always kind of think of the cerebellum as kind of like the human resources of the brain. It doesn't really do the specifics to it, but it makes sure what's being done is being done correctly. All right, let's move up into the diencephalon, which is going to contain your thalamus, hypothalamus, and epithalamus. Talk about the thalamus first. Each side of our brain has the thalamus, which are pairs of, or a pair of oval masses of gray matter, perched at the superior end of our brainstem, beneath the cerebral hemispheres. Nickname for it is the gateway to the cerebral cortex, which is going to be related to that first function that I'll get to here in a minute. Okay. In about 70% of human brains, there's a little bridge that connects the two thalami, I guess I would say. That's called an interthalamic adhesion. You can see that little dot right there is an interthalamic adhesion. Inter, once again, means between, so interthalamic means between the thalamus and between the left and right halves. And there's a vertical Y-shaped sheet of white matter called the internal medullary lamina that divides the gray matter of our right and left sides. It's made up of myelinated axons entering and leaving various thalamic nuclei. Axons that connect the thalamus and cerebral cortex pass through what's called the internal capsule, which you can see here. What is that? I just lost it. Just saw it. No, don't worry about it. It's not that big a deal. I know I saw it in there somewhere, but that's okay. The internal capsule is a band of white matter lateral to the thalamus, and based on their positions and functions, there are 7 major groups of nuclei on each side, and this includes the anterior nucleus, medial nuclei, lateral group, ventral group, intralaminar nuclei, midline nucleus, and reticular nucleus. And that's why these are all shown with various colors. And notice we have different labels for each of those colors. Not something I would ever ask you to know, but those are the different parts of groups of nuclei to the thalamus. As far as function goes, probably its most important role there is the first bullet, and explains its nickname. For most sensory impulses that travel out to our cerebral cortex, they travel through the thalamus first. So they make a pit stop. So that's why it's the gateway to the cerebral cortex. So when you're itchy, when you see something, when you smell something, when you are hot, when you're cold, most of those different sensations that we feel make a pit stop in the thalamus before ultimately traveling to specific areas in our cortex. So obviously it's very involved with sensory function. It's also involved with motor function by transmitting information from our cerebellum and basal ganglia to the primary motor areas of the cortex, relays nerve impulses between different areas of the cerebrum, plays a role in maintenance of consciousness, and then on the sides of the thalamus are several groups of nuclei that are functioning or have functions and emotions, memory, learning, cognition, and then various sensory information, things like auditory impulses, visual impulses, arousal, and olfaction, which olfaction is the sense of smell. We'll talk a little bit more about the kind of specifics to that when we get into learning about what we call our emotional brain, the limbic system, here after a bit. Okay. Kind of in front of and a little bit more so below the thalamus is the hypothalamus, which forms actually a wall of the floor of our third ventricle and is a super, super, super important region of our brain because it controls our autonomic nervous system. And remember, the autonomic nervous system is responsible for controlling our cardiac muscle, our smooth muscle, and our glands. All those things that are done automatically for us, you can thank your hypothalamus. So controlling your heart rate, blood pressure, your secretion of different glands, sweat glands and oil glands, and all those things are just done without even our conscious awareness. It's also the major regulator of our endocrine system, which is super, super important because that's the other major system that is in charge of everything in our body. So your hypothalamus is super, super important for just regulating and controlling those basic survival things. There's 4 parts to it, nuclei that are found in 4 major regions. Mammillary, you have tuberal, which is shown here in blue, supraoptic, these things in green, and then the purple shows what we call the preoptic. There's all sorts of different parts to it, not so much stuff that I would really focus on. And we'll kind of point out the mammillary body. There's actually one on each side, so there's mammillary bodies. These are things that we'll talk about here in a little bit. You can see here's that thalamus, and there's that interthalamic adhesion that I was talking about on that previous page. There's the pituitary that I was talking about, this little thing that connects the pituitary to the hypothalamus called an infundibulum, and we'll get into that more in our next unit. And as far as functions go, it secretes hormones, secretes 9, well, I guess it secretes 7 hormones that are controlling or responsible for controlling the anterior pituitary to do things like regulate growth, metabolism, reproduction, and stress. We'll kind of talk about the relationship between the hypothalamus and the anterior pituitary in the next unit. When we get to the endocrine system, it also produces 2 hormones, but it doesn't secrete them. These 2 hormones are going to be carried to the posterior pituitary, which then is going to be released there. And what I mean by that is posterior pituitary is just an extension of the hypothalamus that releases the hormones that the hypothalamus makes. And those hormones we'll get into, like I said, in the next unit. Other things the hypothalamus does, it's involved with the autonomic nervous system, as I've talked about. Influences heart rate, blood pressure, GI secretion, motility, among other things. Well, if you think back to early in this lecture, I said that the medulla was involved with controlling our heart rate, our blood pressure. Well, medulla is directly involved with doing that, but it only does so when it's told by the hypothalamus. So hypothalamus is truly what's in control. It's your thermostat, so it causes you to shiver when you're cold. It causes us to sweat when we're hot, so it helps regulate body temperature in that way. It does other things to do so as well. It's our food and water intake center, so it monitors things like your blood sugar and your amino acid levels. It makes you hungry if they're low. If our water level, or what we call blood osmolarity level, is low, it'll make you thirsty, so we'll drink more and then become less dehydrated. It sets our sleep and circadian rhythms, which circadian rhythms are our biological clock, not like the biological clock that you hear like people say when they need to have babies because they're in their 30s or whatever. Biological clock, as far as hypothalamus goes, is a repetitive 24-hour cycle of events that occur each day. One of the most important aspects of that is our sleep-wake cycle, but it also involves different things, hormonal changes, even body temperature fluctuations. So it's a little more complex than just being awake and being asleep. It's involved with memory by allowing signals to travel from the hippocampus, an important memory center, to our thalamus, and then it's involved with various emotions. And so we'll get into it a little bit more, especially some of those parts I was pointing out at the bottom of the page to regulate things like anger, aggression, fear, pleasure, contentment, as well as even sexual drive and orgasm. And in this illustration, it's not labeled, but right in here is what we call the pineal gland or pineal gland, which produces melatonin. Melatonin, I always tell students not to confuse with melanin, which we learned early in the semester, is the chemical that our cells in our skin produce that give us, a tan, give us our skin color, give us our hair color, even give us the color of our eyes. Melatonin, however, is what helps set that biological clock, our circadian rhythms. We produce more of it at night, so it's believed to be what helps, and it's kind of a natural sleep aid. In fact, you can buy melatonin over the counter, synthetic forms of it. Well, I just said that the hypothalamus did the circadian rhythms. Well, the hypothalamus is actually in control of the pineal gland, kind of like what I was talking about with the medulla. You can describe your pineal gland as the main structure of the epithalamus, the kind of last major part of the diencephalon. There's one final part of the diencephalon, what we call circumventricular organs, or CVOs, which are found in the wall of the third ventricle that monitor chemical changes in our blood, which is because they lack a blood-brain barrier. This includes parts of the hypothalamus, pineal gland, pituitary, and a few nearby structures. So overall, they help coordinate various homeostatic activities, both the endocrine and nervous systems, like regular blood pressure, fluid balance, hunger, and thirst, which is kind of the most important part, but something kind of interesting and included because in your textbook, the fact that this might be or is at least thought to be one of the sites of entry for HIV into the brain, which can cause dementia and other neurological disorders, unfortunately. All right, on to the biggest, most complex part of our brain, what we call the cerebrum. Largest by far and away, 80%, made up of two halves, what we call the right and left cerebral hemispheres. And we can see, here's a lateral view to show the right side here from above. We can see the two hemispheres, and then we're going to take a little segment out of it to talk a little bit more about the gray matter, white matter stuff. Notice here that it's folded just like what we saw with the cerebellum. The little folds or rolls, as I kind of like to think of them, are called gyri, which is the plural form of gyrus, and little indentations are called sulci, which is the plural form of sulcus. So that is going to increase the amount of surface area and thus more of what we call the cerebral cortex. When people talk about their brain cortex, they're typically not going to be talking about their cerebellar cortex. Instead, they'd be talking about the very thin shell, about 2 to 4 millimeters thick, of the cerebrum, which you can see here in our illustration. It's gray matter, and that's where your highest level processing takes place. And this is what separates us, especially the fact that we have such a big cerebrum, from lower animals. It's what gives us our intellect and our personality. So what allows us to compose music and plan for the future and do all those things that your lower level animals like your dogs and your cats just really don't do. As far as other things, each of the hemispheres is divided into 4 lobes. They're named for the bone that overlies them. A frontal lobe is the front, and the parietal lobe, which would be deep to the parietal bones, occipital lobe in the back, and then on the sides would be your temporal lobes. Okay, each is separated by a specific what we call sulcus. Most prominent of those would be a central sulcus, which runs right here between the the frontal lobe and the parietal lobe. There's also a parieto-occipital sulcus between the parietal and the occipital lobes. And then there's a lateral cerebral sulcus, which is below, or between the temporal lobe below and the frontal and parietal lobes above. So that's what runs right through here. Kind of the only of the sulci that have specialized names. There's also a very prominent what we call fissure that runs right down the middle of the cerebrum and separates the two hemispheres from one another, what we call the longitudinal fissure. There's another fissure, what we call the transverse fissure, and that's what I mentioned earlier, separates the cerebellum from the cerebrum. I want to point out right in front of the central sulcus is a region of our frontal lobe called the precentral gyrus. And then right behind in the parietal lobe is what we call the postcentral gyrus. And I'll talk about those on the next page. And that's because they're involved with some very, very, very important things. Okay. There is a fifth part associated with each cerebral hemisphere. It's kind of the fifth lobe. It's shown here kind of transparently, what we call the insula, and then notice here in parentheses it says projected to surface, just because you would have to kind of separate and pull parts of the brain out to actually show it. So it's kind of like the ventricles I was showing you earlier. They're just kind of showing where it would be located at in kind of the retrospect of the brain. Okay, let's talk a little bit more about the cortex. Very, very important. So let's read through. Specific types of sensory, motor, and integrative signals are processed in certain regions of the cerebral cortex. Generally, sensory areas receive sensory information and are involved with perception, which we've learned in our first lecture is that conscious awareness of sensation. Like when we are aware of what we're hearing, what you're seeing right now, what we're smelling, Dr. Pepper that I'm drinking, I'm tasting, I'm consciously aware of it. Things like when we're hot and cold, itchy, and all those things. There's also motor areas of our cortex that control the execution of voluntary movements, like what I just did with scratching the back of my head, involved contracting skeletal muscles purposely, voluntarily. And then there's also association areas that deal with more complex integrative functions, things like your memory, your emotions, your reasoning, your will, judgment, personality traits, and intelligence. And like I said, this cortex and your large cerebrum are super, super complex and what separate us from, your lower level animals. There are many, many, many functional areas, and that's illustrated here with our picture with all sorts of different numbers and things labeled. I've kind of picked out some of the most important ones that I felt like you should know. I kind of was already talking about two of them. I just really didn't get into it. In your postcentral gyrus, so right here behind that central sulcus, this general kind of purple shaded region is what we call the primary somatosensory area. That's where the sensations that come from your skin and your tendons and your joints ultimately end up going. So when you're hot, when you're cold, when you're itchy, when you're tickled, that kind of stuff, those signals are going to ultimately end up traveling here. Okay. In the precentral gyrus, this general red area is what we call the primary motor area. And that's where signals originate, enabling us to contract our skeletal muscles in that voluntary manner that I was talking about earlier. Okay. Other things, primary visual area. This is going to receive visual information as involved with visual perception. So we're using that right now. Process what we see. It's found in our occipital lobes, mainly on the medial surface next to the longitudinal fissure. Primary auditory area, you're also using that. It's involved with sound and auditory perception. Found in the superior part of the temporal lobe near the lateral cerebral sulcus. Gustatory areas involved with taste and taste perception, because gustation is taste, as well as taste discrimination. Found at the base of the postcentral gyrus. Olfactory areas involved with smell and is involved with olfactory perception. Found near temporal lobe on the medial aspect. I already mentioned the primary motor area. A couple kind of specialized ones. Wernicke's area interprets the meaning of speech by recognizing spoken words. You guys are doing that, using that right now. It's found in a broad region in the left temporal and parietal lobes. I'm using the Broca's speech area because I'm articulating. I'm forming words. Found in the frontal lobe, close to the lateral cerebral sulcus. About 97% of people, it's in our left hemisphere. And then a very broad area in our frontal lobe, right kind of deep to our forehead, is what we call the prefrontal cortex. This is involved with your personality, your intellect, your memory, all sorts of different things that I'm not going to just read through. Unfortunately, being it's located in our forehead, it's kind of susceptible to damage. Like when you have a car accident, sometimes your head hits the steering wheel or the dash. And it's not that uncommon for people to have memory issues or even completely be a different personality after having that traumatic brain injury to that prefrontal cortex. As far as the image goes, I wouldn't ask you to know specifics to it. It's too complex, but pretty cool. Now keep in mind, that was just the very thin shell on the very outside of our cerebrum. Beneath that is a very large interior made up mostly of white matter and what we call tracts, which we learned are myelin axons that can do 3 different things. Okay, when we talked about tracts in the spinal cord, we talked about, and I kind of use an analogy of an elevator carrying signals up and down. We do have those in our cerebrum. We call those projection tracts. We can see those here in this view. You can see them here going up and down. You can actually even trace down where they crisscross, decussate, and arm medulla oblongata. Okay. There's also commissural tracts, which enable the 2 hemispheres to communicate with one another. Okay. Commissural tracts, I always tell students, think C for commissural, C for cross, and they form a structure that's called the corpus callosum, C for corpus, C for callosum. And if you take the page and you turn it sideways, that structure, which I pointed out on that very first page, actually looks kind of like the letter C. So that's kind of cool. Back when it wasn't illegal to do brain lobotomies, they actually used to snip this and prevent the 2 hemispheres from connecting. And that's where a lot of research that went into like right brain and left brain functions come from, what we call hemispheric lateralization, stuff I won't really get into. The last thing I'm going to talk about are the kind of ones that kind of look like a rainbow to me. These are called association tracts. What they just do is they allow a signal maybe to go from the front of the brain more to the back of the brain or vice versa. So the key with those is they stay in the same cerebral hemisphere, which is different than the commissural tracts. Okay, so that was white matter. We got gray matter again, what we call basal nuclei. Previously, they were termed basal ganglia. And I don't like the fact that they used to be called ganglia, and I'm glad they changed it, because as we learned, ganglia are masses of gray matter found outside the central nervous system. Well, nuclei are found in the central nervous system, so that makes a lot more sense. These are found in the very deepest parts of the cerebral hemisphere. There's 3 parts, or 3 nuclei: the pallidus, globus pallidus, the putamen, caudate nucleus. Together, the globus pallidus and putamen are 2 basal nuclei, just lateral to the thalamus, and together they're the what we call lentiform nucleus. Okay. You can see kind of their location. There's the 2 lateral ventricles. Down here would be the kind of area of the thalamus. And you can see the structures that make it up, kind of surrounding it. Same thing, kind of right in there. The caudate nucleus has a large head connected to a smaller tail, a comma-shaped body. Together, the lentiform and caudate nuclei are called the corpus striatum. Major function of the basal nuclei is to help regulate and initiate terminate movements. Globus pallidus helps regulate muscle tone required for specific body movements. Basal nuclei are also going to control subconscious aspects or subconscious contraction of skeletal muscles, which is something that we're doing, like running up and down our spine, even though we don't have to have, we've talked about how like we have to have conscious awareness, and I'm purposely doing this with my hand and that kind of stuff. We're also subconsciously contracting muscles without thought going into it, contracting our diaphragm to breathe, spinal muscles that keep our head and neck and our spine vertically aligned and that kind of stuff. We don't have to consciously think of those because of activity of our brain, and basal nuclei is involved with that. Basal nuclei also help initiate and terminate some cognitive processes such as attention, memory, and planning, and they may also act with the limbic system to regulate emotions. Speaking of the limbic system, let's talk about that right now. This is a ring of structures on the inner border of the cerebrum and the floor of the diencephalon, encircling the upper part of the brain stem and the corpus callosum, and several parts make it up, and they're all shown kind of here in green. I've always kind of told students think of this as like the primitive brain, the brain that controls more lower level animals, emotions, doing what makes them happy and feeding their basic desires, but not what should control everything. And what we do, because we have the ability to kind of more plan for the future and think rationally and that kind of stuff. Hopefully we do anyway. Okay. Parts that include or are included in the limbic system: cingulate gyrus, which lies above the corpus callosum, regulates emotions and pain, also involved in predicting and avoiding negative consequences. Hippocampus, I mentioned earlier, is a portion of the parahippocampal gyrus, involved with memory. Dentate gyrus lies between the hippocampus and the parahippocampal gyrus. Amygdala composes several groups of neurons located close to the tail of the caudate nucleus, involved in fear and discrimination of objects necessary for human survival. Mammillary bodies, I pointed out earlier, parts of the hypothalamus, serving as relay stations for reflexes related to the sense of smell. Two nuclei of the thalamus, anterior medial nucleus, olfactory bulbs, which are flattened portions of the olfactory cranial nerves, and are therefore involved with olfactory pathway, the sense of smell. And then the fornix striae terminalis, striae medullaris, medial forebrain bundle, mammillothalamic tract are linked by bundles of interconnected myelinated axons. Nickname for this is our emotional brain because it governs emotional aspects of behavior, things like pleasure, pain, docility, affection, fear, and anger, as well as even olfaction or smell and memory. So very kind of innermost parts of our brain. Pretty complicated image, but there's a lot of good things here labeled. Some we've talked about and pointed out, some that are more brand new to us. Okay, so that summarizes here with this table 14.2 all the different parts of the brain. This is a figure from or a table from your textbook. I included it because it's kind of an easy place to look and see what happens where. Not something I specifically pull stuff from, but something you might use to help you study and learn them. Last thing I want to talk about before I wrap up are the various cranial nerves, which I've mentioned at several various points in this unit. Nerves of the peripheral nervous system, originating from or terminating in the brain. 12 pairs of them exist, and they're numbered from anterior to posterior with Roman numerals 1 through 12. And there's 3 types, which is a little bit different, quite a bit different actually, than what we saw with spinal nerves that we talked about in our last lecture. If we think back to our last lecture, we learned that spinal nerves were like a 2-lane highway, which meant that signals were going in and out in opposite directions. That's how some of our cranial nerves are, but not all of them. Okay? That would be what mixed nerves are. Okay? Whereas sensory nerves, there are a few of those that contain or predominantly contain sensory neurons, which means that it's a single or 1-lane highway carrying signals into the brain. Whereas motor, the opposite, okay, contain or predominantly contain motor neurons and aren't going to carry signals out, and therefore are involved with controlling skeletal muscle or some type of muscle movement. Okay. I've included kind of a term, or not term, but a phrase to help you remember them. There's other ones out there, but notice I've bolded and capitalized the first letter of each word to give you the first letter of each of these different cranial nerves. Okay. Listed here are the cranial nerves in order from 1 to 12. Okay. Gives you their real name aside from their number. Gives you what they are, motor, mixed, or sensory, and it gives you nicely, very briefly, description of what their function or functions are. Okay. And then even at the bottom, here's another example of a mnemonic device. Okay. I do expect you to know them, the functions, the type, the names. So the table would be very important to study. As with this illustration that shows them on the left side. Now, keep in mind that these are just like the basic trunks of these nerves. For example, like the vagus nerve on each side. Vagus nerve is a nerve that travels down through our neck, into our chest, and into our abdomen. And it controls a lot of different parasympathetic responsibilities. We can only see a little portion of it because it's been cut away, like a tree that's been cut down. You can just see the stump that's left over. So think of these as like the stumps where they're connected to the floor or the ground, in this case, the bottom of the brain. This is that same illustration that I've shown you a couple different times. As far as what you should study, just the 12 nerves on the left. And then I've already mentioned this little fattened portion, the end of the olfactory cranial nerves is called the olfactory bulb, which is something we'll talk more about in our next unit. All right, that wraps us up. Thanks, guys.